Hello everyone, welcome back. The next speaker of the session is Dr. Vidyanand Nanjundiah from Center for Human Genetics. He'll be talking about unicellular organisms and the evolution of social behavior. So let's hear it from Dr. Nanjundiah.
Thanks, Asha. What I'm going to, is this okay now? No. Okay now? Better? No? Now? Social behavior? Okay, you got it. So as I said last time, what I'm going to talk about is really going to be on the evolution of multicellularity, but as you will see, there are good reasons for pretending that it's also about the evolution of social behavior, because the two of them mean the same thing in this context. But the narrow theme I'm addressing is a possible evolution, possible route for the evolution of multicellularity. I should also say that what I'm going to do here is based on a piece of work, a survey, really a review carried out with Iñaki Ruiz Trillo and David Kirk, where we considered 3 different systems, mine being the cellular slime molds or Dictyostelium. You saw the name last time and you see it again. They contributed respectively on what are called the phylasterians, Captospora, and on the green algae, the Volvocalis or Volvox and its family. I'll make brief comments about the other 2 at the end of my talk to show that there is a set of parallel themes running through. Otherwise, my talk will be pretty much restricted to the cellular slime mold. So that's why I call that a case study. Not very clear when multicellularity evolved. What we are sure about is that it did evolve in the sense that life on Earth was unicellular for a long time. In fact, consisted of cells without nuclei for a long time. Then you had the evolution of cells with nuclei. And from then on, at some stage, you had the evolution of cells with nuclei grouping themselves into multicellular units. Interestingly, a parallel evolution occurred among non-nucleated organisms, among the ancestors of today's bacteria. And the parallel evolution also led to the evolution of multicellularity, aka social behavior, in a group called the myxobacteria, but I will not deal with that, I'll just mention it briefly. So this talk is going to be concerned with the evolution of multicellularity in ultimately in animal lineage. And as far as the present day animals are concerned, their unicellular ancestry appears to date, going by a whole host of work by paleontologists and climate, people who study paleoclimate and so forth, seems to date back to something like 650 million years ago. And Andrew Knoll, who has done a lot of study on this particular aspect, dates it to the time when the atmospheric oxygen level saw a steep climb. So it's somewhere along the middle of the gradient of rising oxygen concentration, which in turn was probably triggered by an increase in the number of cyanobacteria, blue-green algae in marine waters. So that's the time we're thinking of. In general, there are 2 routes to multicellularity that people talk about. One is a number of things which come together. That's the picture on the left-hand side, aggregative multicellularity. And the other is a picture in which you have one kind of thing which makes many copies of itself which stay in place, which of course is the sort of multicellularity we are most familiar with. There are modifications on these 2 very general themes, and we'll probably see a couple of modifications towards the end. But one of the purposes of my talk, and this was prefigured by what Stuart Newman said yesterday, one of the purposes of my talk will be to say that maybe aggregative multicellularity might have been the way through which we got the sort of multicellularity we are more familiar with now, which is the division of a fertilized egg today. We'll see whether that can be made to look plausible at all. Right. There are many, in evolutionary biology, especially when you are dealing with macroevolution, with major changes in evolution, you have a whole lot of interrelated themes, and it's not obvious how to factor them out. This is a disadvantage of thinking of macroevolutionary problems, which you don't face generally when dealing with microevolutionary problems, where you can look at a single gene or a single protein in controlled conditions, ideally in the lab, and can pose precise questions that you can answer by experimentation. So here the two themes are a unicellular individual becoming a multicellular individual, which I've already mentioned, and also that of asexual reproduction having given way to sexual reproduction. And the reason these two themes are, both of them of course are enormously big steps taken in evolution. The reason these two themes are intertwined is because the familiar form of multicellular development that we know today is pretty much inextricably tied up with sexual reproduction. So that's what's written there. So you have a multicellular individual which through this very complex process called meiosis produces some cells with half its own genomic complement, and then two such cells with half the genomic complement come together, reconstitute in some way the parental type, though as we know it's not identical to the parent, and then this divides many times and gives you the multicellular individual, and you come back again. So the cycle closes upon itself. So this is meiosis, if you wish, and then fertilization, growth, and then of course meiosis in the next generation. The modifications on this theme, and I just mention them here because one should be aware of them, not that I'm going to treat them here. You can get parthenogenetic development. So you have multicellular organisms essentially like us, so let's say vertebrates if you wish, where you have no meiosis at all, but females can give birth to essentially the progenitors of what's going to become their daughters. So diploid eggs if you wish, which are fully capable of developing. You can get budding in certain organisms, followed again by growth and increase in size. And budding is also sometimes accompanied in these systems by fusion, where two small buds get together. And then finally, you can get propagules made by the multicellular individual, technically different from budding, but in principle the same. So some part of the multicellular individual that separates, and in this case we are thinking of a single cell separating, and that part which separates now dividing many times and so forth, and the multicellular individual being regenerated by aggregation. So this is the theme that we are going to try and follow. What's interesting is that you can get combinations of these circumstances occurring in the same group, and sometimes even in the same kind of organism, depending on the environment and so on. Why this interest in aggregative multicellularity? Well, it turns out that if you look at the best available phylogenetic tree today, based on 18S RNA sequencing, deep sequencing, you end up with these large groups, which are now called supergroups of eukaryotes. So these seem to be the largest subdivisions into which all eukaryotes fall. And they're the ophisthocorns, excavates, archaeplastids, which are the ancestors of the green algae and the plants of today, alveolates, straminophytes, rhizaria, and amoebozoa. So there are 1, 2, 3, 4, 5, 6, 7 such supergroups. Turns out that except for the archaeplastids, each of these supergroups contains members today in which you get aggregative multicellularity. So this is something which has arisen independently in evolution in at least 7 of these different lineages, and because of how ancient this phenomenon is, we think that it may well have arisen at roughly the same time. The archaeplastids, so the group which comprises the green algae and the plants, is going to be represented in today's talk by the Volvox family, as I mentioned, and there it's clearly a form of colonial multicellularity that you get, this cell dividing into many cells and forming the group thereby. But interestingly, there are circumstances in which a member of this group, in fact, in some sense, the most primitive member of this group, the cell called Chlamydomonas, also shows aggregative multicellularity under certain circumstances, which may or may not be natural, but it's interesting that it does so. So this is something which cells, and it's going to be a theme of my talk, cells seem to be predisposed to doing if given the right conditions. So this is again the same kind of tree that you saw previously, but this time to illustrate that in each of these separate major branches, you've got single cells coming together, and not only do they come together under certain circumstances, they finally end up building what looks like the same sort of multicellular body or differentiated structure. From its rough resemblance to the fungal fruiting body, we saw this yesterday, this is also called a fruiting body. So all of them form fruiting bodies, and the common characteristic of these fruiting bodies is that they rise above the soil, so these are generally found in the soil. By the way, also on animal dung, that's a common rich source for a great many of these organisms. But if you find them in the soil, they rise above the soil. The typical height here by microorganism standards is impressive, about a millimeter, can go up to 2 or 3 millimeters. And right on top, what you see in some cases as a ball is a mass of these units which have come together, but which have sporulated. And the reason they have sporulated is because this entire business of coming together in these cases is initiated by starvation. So they are starved, they are no longer able to find food, then they come together and build these multicellular bodies. Below those spores, you have what looks again like a stalk of a yeast or some other fungal fruiting body. This is also called a stalk. The nature of the stalk varies, and that's going to be something I'll mention presently. In some cases, it's a cellular substance. In other cases, it's not. When I say cellular substance, I mean it consists of cells. In other cases, it's not. And when it consists of cells, in some cases, the cells are dead. I mentioned this yesterday again, showing a clear-cut division of labor. And in other cases, the cells are viable, showing division of labor without reproductive division of labor, meaning all cells can, in principle, reproduce in the next generation, as the spores do. But the stalk cells being closer to the ground are presumed to be less effective at reproducing than the spores. And the reason for that is the following. This is a long-held belief and probably true, which is that the evolutionary basis behind the success of this lifestyle is that if you are very tiny and if you are starved, what you want to do more than anything else is to get away from here to some other place. And therefore, rising above the soil offers you a good chance of passively dispersing. Any passing insect, which is usually hairy, or a nematode worm, which is going past, can either entrap you within its hairs, totally unintentionally. Nematodes, it turns out, can also eat these things, but once they are spores, they are unable to digest them, so deposit them later wherever they go, or by pure chance get some of the spores sticking somewhere. And then something rather curious can also happen, which is that if this is a grassy patch, it can be grazed by a whole host of animals, and then a great many of these creatures can get churned up inside the stomach of, let's say, a ruminant which is going around there, or even any other herbivore. And then when it passes dung, it can be laid down as spores once again somewhere else, because the spores once again are not digestible. So it's a rather curious kind of thing. And it turns out also that in addition to insects and worms and herbivores, carnivores can also act as agents of dispersal. So a student here working with me discovered that these things are quite common also in the dung of a tiger and a leopard. And there the guess is, some of the cats, of course, also can graze occasionally, it's known. But in addition, it's possible that via eating meat, they get hold of, eating the meat of herbivores, they get hold of some of these. In any case, so this is a tactic for dispersal which seems to work rather well and has served these people. So comparative similarity. Again, the same sketch as before to show you 4 examples, and there are 3 major groups illustrated here, the supergroups, the amoebozoa, the excavata, and the ophistoconta. And in all of them, you see roughly similar morphology following starvation, amoeboid cells in every case, important point, coming together and building this fruiting body. And then various things happen, as I said, in the fruiting body. So there are 2 problems here that confront us when we look at the system. I mean, you could call them an engineering problem and a biological problem. The engineering problem is, how do these cells come together and build this 3-dimensional structure? So it's a kind of mechanical engineering cum civil engineering problem. The biological problem is, what patterns of gene expression perhaps are implemented by these organisms so that this end point results in a stable division of labor in one way or the other. In other words, what makes them cooperate in the way they do by coming together and building this fruiting body. There are 2 broad answers to this question, and we've been listening to these for the last 2 days now. One is that in some sense, each of these forms of development is a special purpose solution to the common problem of living in the soil and requiring dispersal, so that each step in this detailed life cycle that you see has evolved by incremental genetic change, followed by natural selection in case the change was advantageous, and gradually, step by step, you had this pattern building up. And then, of course, over a larger time scale, you could get, in the phrase used by Darwin, descent with modification, maybe slight variations, and we'll see that variations are also common in this thing. The second point of view, of course, which we've been exploring here in this class, is that in some way the kinds of generation of shape and form that you see here are a consequence of pre-existing physical and chemical properties that these units possessed before they became multicellular, in other words, before the evolution of multicellularity. Following the onset of multicellularity, which was triggered perhaps by an environmental trigger or even by things like population density, viscosity of the medium and so on. Following that, natural selection acted after the change had occurred to stabilize the change. So these are 2 at least conceptually distinct points of view, and perhaps there are elements of both. Must be kept in mind, of course, that what we are seeing now is after these 650 million years have passed, or 500 odd million years have passed. So you can be pretty sure that whatever you find in present day living organisms must have been the consequence of a great deal of evolutionary change after those 500 million. So in some way we are trying to take an eraser and rub out this change and try to see whether we can guess what had happened at the beginning. Okay, so let me very quickly take you through this Dictyostelium life cycle. This is a series of films by K. Inoue, and this depends on luck maybe. How do I? Let me see if it works if I go to the screen. Is it doing something? You are right, though. Okay, yeah, we'll try and go to the copied versions of this. Is it showing something? Now maybe. So what that's supposed to be, this is a bit frustrating. What that's supposed to be is an amoeba emerging out of a spore. So that's the conventional beginning of the life cycle. And these are amoebae moving around, feeding on bacteria and dividing by mitosis. So this is the growth and cell division phase. When that is completed and they begin starving, they communicate to each other via these really rather pretty oscillatory wave propagations, which I think I mentioned briefly last time. And after the period of wave propagation is well underway, they also begin active movements towards common collection points. So this is aggregation. After aggregation, the entire mass falls to the ground and begins migrating. And what it does actually is not to migrate on the soil, but to migrate through the soil until it reaches the surface. And having reached the surface, it goes through this final step, which is to arise on top of the soil. So this is a rather nice example of self-organization within I apologize. This is uncanny. The film worked in my room this morning. When I came and tried it out here, it didn't work. So I downloaded it again here. It didn't work again. So, okay, let's hope it works from now on. So this is simply a repetition of what we saw earlier with an added thing below to show that there is a non-obligatory sexual phase in all of this. And one big unanswered question is, can one ignore the sexual phase, which is what one does usually when talking about the life cycle, or does it in some way play an essential role in features of the life cycle? Okay, so come again. Oh, that happens under what to us working in the lab is a set of very strange conditions, extreme conditions, which is the case, by the way, with most microorganisms. So in this case, you have to have very intense light, flood your petri dish with water, and then pray. And then sometimes these cells form zygotes, which is what are called macrocysts. Doesn't fit there. So the usual life cycle is completely, what we think of as the usual life cycle is completely asexual. Right. So there are ways in which one can study this coming together, the aggregative part. There are people who have built and worked on mathematical models of this. And broadly, the models fall in two categories. There are so-called continuum models, which are similar to the Turing reaction diffusion kinds of models that Newman talked about, except that here you have cells or amoebae, so it's their concentration, if you wish, or their local density that's represented there. And then you have an attractive chemical, a chemoattractant made and released by the cells themselves. It's their concentration that's being, whose rate of change is being monitored here in the second equation. And the broad principle of the equation says the amoebae can diffuse, so that's a diffusion term, and in addition they can move up chemical gradients, attractant gradients, which is why you have a negative term there. The chemical itself can diffuse, of course, ordinary diffusion. In addition, it can be produced by these cells, so that's a production term, and it can be degraded by these cells, there's a degradation term. So the underlying system is very simple. What is interesting is that it gives rise to a fair amount of complexity, which resembles what you see in the real system. Equally interestingly, there are some things that it does not give rise to. And one of the most important things, at least within the scope of this simple two-component model, it does not give rise to the oscillations that we saw a bit earlier. Remember these periodicities that were shown both by wave propagation and during aggregation. That cannot be mimicked by this model. However, the oscillations result quite well when you look at a discrete model, which is a single cell model, because here you begin by modeling the oscillator itself within a single cell, and this was really pioneered by Albert Goldbecker, who you will listen to later, and the model here is really extremely simple. The signal in the simplest case we know is this molecule called cyclic AMP. It's made inside the cell from what's called ATP, and that's representation of the enzyme that makes it, an adenyl cyclase. Cyclic AMP, which is the chemoattractant, goes out from the cells, and interestingly, there's a positive feedback element here. Cyclic AMP, having gone out, can bind to a receptor. That's a second different molecule outside the cell, and binding to the receptor activates cyclase. So you see this positive feedback loop there, right? And if that loop, which of course by itself would lead to an explosion, if that loop can be shut off by something or the other, and there are any number of things you can do that, including enzymes, enzyme desensitization, and so forth, then you can see that the cyclic AMP levels will shoot up, crash down, shoot up, crash down, and you get a natural periodicity which gives rise to the oscillations. Now you can combine that, so in a combined kind of model, and I think Stuart Newman used the word morphodynamic. So you can build a morphodynamic model in which you get oscillations and wave propagation and cells coming together. So this can be done rather satisfactorily, and you can show, we'll come to the three-dimensional shape a while later, you can show that these things having come together can build a multicellular mass, right, which is there. So you're there at least at the first part of what you wanted to do. So what have you done so far? You've shown that if you have cellular units which are able to stick to each other in some way, which are able to signal to each other, and which move by ordinary amoeboid movement, then they are capable of coming together, forming one mass. And the reason they do that is because whenever there are local concentration fluctuations of these cells, the fluctuations build upon themselves. And the reason why they build upon themselves, you saw in the previous two slides. There are positive feedbacks, A due to chemotaxis, and B due to this oscillatory loop which strengthens the signal going in. Right. Having come together, this is the mass that they build, which keeps migrating. This is called the slug. You saw that a while ago. This is the final structure that they build, which is called the fruiting body. Now, something rather nice you can show is that precisely as in higher animals, there are other nice coordinated spatial and temporal patterns of gene expression which you can see in the slug, which mirror what those amoebae do when they form the fruiting body, and this shading here shows you what they do. So the amoebae in the back of the slug form the spores eventually. The amoebae in the front of the slug give rise to the stalk eventually. And because, at least in this particular species, you have reproductive division of labor, namely the spores can reproduce, whereas the stalk consists of dead cells, the analogy has been made to the germline and soma of a multicellular organism, the germline being capable of reproduction and the soma not reproducing. And the argument has been made that this whole business of coming together, including the seeming self-sacrifice of the stalk, because these are dead amoebae, remember, this whole thing coming together makes sense because all the cells here are clonally related to each other. They are members of the same clone. So this so-called altruistic behavior of the cells that sacrifice themselves can be explained by saying that they are helping the reproduction of the remaining cells, which are spores, which have their own genes. So this is the verbal argument given for why these cells come together, behave collectively, and exhibit division of labor, through which some of them are advantaged and others are disadvantaged. Because the argument is that the disadvantage is only apparent. At the genetic level, they are as advantaged as anyone else having the same genes. But it must be remembered that all of these conclusions were carried out in the lab, and this was done by microbiologists, continues to be done by microbiologists. And one of the articles of faith in microbiology is that you work with pure cultures. You don't like contaminants. So the first thing you do almost before you're beginning an experiment is to do a subclone and then work with that subclone. So intentionally, these experiments were done with clonal cultures. Unintentionally, they led to one thinking that that may have been a factor behind this sort of behavior. There were other reasons too, which I will come to later. Now, there are a number of things which complicate this simple-minded picture or make it more interesting depending on your point of view. One of them is that cells are heterogeneous, and this heterogeneity is a spontaneous heterogeneity which arises in a cell mass. So this is an experiment in which you have a mass of cells, in this case all members of the same clone, whose calcium levels are being monitored, and you can see clearly that the calcium levels follow a bimodal distribution. There are some cells with a lot of calcium and other cells with little calcium, clearly two different modes. Turns out if you put these cells together after secondarily labeling them, the cells which had a lot of calcium end up in the front of the slug. These are the same pictures, by the way, bright field and fluorescence. Whereas the cells which had very little calcium end up at the back of the slug. And we've already seen from the fate map that we saw a while earlier that these are the cells that are fated to die. These are the cells that are fated to form spores. So you see immediately that even before the cells come together, this is at this single cell stage, even before they come together, there's some sign in these cells of what their future fate is likely to be. It's unlikely to be a one-to-one correspondence, but you can certainly see that there are biases in what they're going to do. So what this tells us is that heterogeneities between cells are a strong factor in deciding on what happens later. And these are heterogeneities of phenotype present in a common genotype. So you can imagine that if genotypes also varied to begin with, if these were not members of the same clone, the heterogeneities would get accentuated, would get sharpened. Now, once you put these heterogeneities together, in other words, assume that your starting population is not uniform, you can carry on with the same game that I showed you a while earlier, and this is a whole series of simulations carried out by Pauline Hogeberg and her students. And you can in fact get very impressive looking three-dimensional masses too. So you can model the entire life cycle based only on the assumption that you have amoeboid units capable of movement, capable of sticking, capable of attracting each other because of these chemotactic gradients. And then because of heterogeneities in precisely the properties that I mentioned. So for example, heterogeneities in adhesiveness would make them salt out and give you some cells which are shown here as green, some as red, some as blue, dark blue and light blue. And I go back again to the slide which was shown by Stuart Newman yesterday. Sorting out of cells, in his case he used surface tension. In this model, adhesiveness was used as a parameter. You can use surface tension as well, it's been done. So broadly speaking, this heterogeneous mass of cells, given these elementary properties, can recapitulate at least the interesting morphogenetic features of the life cycle. Yeah, please. So the question is, when they move, are the cells which are going to form the stalk later and die, are they the ones in the front and are the others in the back? Yeah, the answer is yes. Okay, so you have so far differences in properties between single cells, which at least in a rough way can correspond to what you see in terms of the development of form. Now it turns out that you can also ask whether interactions between cells are important for this purpose. And for answering this question, some years ago, Ashok Bhogle used a rather interesting parameter. And the parameter was the relative proportion of stalks to spores. You remember we talked a little bit about allometry yesterday. This particular ratio, the proportion of stalk to spores, seems to be isometric. In other words, it scales with the total number of cells. And in fact, it scales over an amazing range of total numbers over 4 or 5 decades of cell numbers, which means going anywhere from about 10 cells, even though that's pushing it, certainly 100 cells, to about 10 to the 7 cells. You get a constant proportioning of stalk and spore. Therefore, as a parameter in itself, this is not very useful to distinguish between models because all models are tailor-made to make sure that you get constant proportioning. But you can play with this system by going down in the total number of cells that you have and going down to, if you can, a handful of cells. In these experiments, 20 odd was the starting number. And when the numbers are so small, it turns out that you get detectable fluctuations. So these are a measure of what's called developmental noise. And you can use these fluctuations, which are measurable, quantifiable, as a discriminating tool to distinguish between models. So you can construct a whole lot of models of cell-cell interactions in which the ratio of cell types, the mean ratio of cell types remains constant. So in some sense, the signal remains constant. But depending on the details of the model, the variation about this mean, what you might call the noise in the signal, varies, and use that to discriminate between models. And it turns out, so these are the actual data here, and these are three kinds of models: a purely stochastic model with size sensing incorporated, with many other things incorporated, switching and feedback. And it turns out that this sort of thing, you can even see this very roughly by eye, this sort of thing fits the actual data much better than anything else. By the way, I should tell you what the data is. I forgot to do that. On the x-axis, you have the total number of cells, and I've told you it varies from whatever, 100 to 10 to the 7 easily. But here the range is only from about 20 to 100, 20 to 120. So you're in the small number range. And the y-axis is the proportion that we want to measure, which is the relative number of spore cells to the total number of cells. And that actually flattens out here at something like 80%. So this curve, which rises from here, goes to 80% and becomes flat there forever. So 10 to the 7 is somewhere there outside the room. Okay, but it's this region that we are looking at now. And here you can discriminate between models. And it turns out now that the models which allow you to fit this data best are the ones in which you have the following things. So I'll just come to the final model, the most successful model. You have an amoeba. The amoeba is able to spontaneously differentiate either in the spore direction or in the stalk direction. And our best guess at this time is that this is a purely stochastic process. So there's something within the amoeba which does this. We have correlates of whatever might be behind this stochastic process, and the calcium level, as I showed you a while ago, could be one of those correlates. But these tendencies, spontaneous tendencies to form spore or stalk, are overlain by other factors, among them trans-determination. So there's a spontaneous probability that a spore-like cell, or a pre-spore cell as we call it, could become a pre-stalk cell, a spontaneous tendency for a pre-stalk cell to form a pre-spore cell. And on top of that, there are strong negative feedbacks from this to this transition, and from that to this transition. So those are the thick arrows with the bars, thick lines with the bars at the end. So you have a rather complex process here, which best fits not just the mean outcome of patterning, at least this aspect of patterning, which is the proportioning problem, but also fits the variations that we get in this mean, and the variations are visible only when you are at very small numbers. And this complicated model involves A, cell heterogeneities, which are reflected in these stochastic probabilities of becoming a spore or a stalk. B, interconversion of cell types before they are fully differentiated. C, negative feedbacks which stabilize cell types when they are differentiated. And D, size sensing, which is shown by the fact that this thing ultimately saturates at 80%, but begins from a very small number. So the problem, in a sense, for developmental biology or developmental genetics is, first of all, to see whether these things add up, and secondly, to see what the molecular underpinnings of these steps are. And we have gone some way in this direction, though I'm not going to discuss any of that now. It appears as if this is a reasonably robust model to fit the overall picture. Okay. Now coming back to the situation that we really want to describe, which is what's happening in nature. Now things get even more complicated here, because here it turns out that what we called stochasticity in terms of a single cell taking a decision to become a spore-like or a stalk-like cell, this uncertainty, if you wish, in decision making is built upon many other layers of uncertainty. So for example, I've already said that when these amoebae are starved, they come together. It turns out that that's not strictly true, and this was shown by Daria Dubravcic in Clement Isak's lab some years ago. Turns out that there's heterogeneity here as well. There are some amoebae which, so to speak, seem to prefer to take the risk of not joining other amoebae, remaining single, presumably so that if food reappears soon, however, then they're ready to feed and again get going. So they gain an advantage, presumably, over the rest. whereas the bulk of them do come together. So there's an uncertainty there here. So when food runs out, I've told you that they aggregate, but many of them can remain as an amoeba, not join the aggregate. Some of them, in fact, can differentiate to form a small cyst-like structure called a microcyst, and this is very common with soil microorganisms, especially other soil amoebae, which are not part of this at all. They encyst themselves, so this can wait out starvation, but as a solitary cell. If they aggregate, of course, yeah, I've already said that there can be sexual cycle which have completely ignored. On the other hand, the asexual cycle, you can get this slug, you can get fruiting body.
Excuse me, sir. Sir?
Yeah.
So the amoeba which forms the fruiting body, are they heterogeneous or homogeneous?
Well, as I said, in the lab, they can be genetically homogeneous, but phenotypically they're heterogeneous. In nature, as I'll tell you in a moment, they're genetically heterogeneous and also phenotypically heterogeneous. One point to which I want to direct your attention is that the probability of a cell dying through all of this, through all of these alternative life cycle strategies, if you wish, that probability can vary a lot. So in case food runs out and an amoeba remains a single amoeba, then you have a variable time-dependent probability of death. We can't quantify this because we don't know what the chances are in nature that food will recover. On the other hand, if it becomes a microcyst, then presumably the probability of survival in the long run is pretty high because it has encysted itself, it's resistant to the environment. But how high it is, we don't know. And we don't know because people don't work much with microcysts, how the microcyst itself degrades with time. So this we don't know. The sexual cycle is rather interesting, and this throws up once again this paradox which is at the heart of the evolution of sex, which we are not discussing here, which is that the sexual cycle is an enormously costly exercise. People commonly talk about what's called the twofold cost of sex, which is really a twofold cost of meiosis, the cost of the reduction division. The size of the genome reduces by half. But here, the cost can be as high as 99%. Even though this is a different kind of, completely different kind of system. And the reason it can be 99%, and I didn't mention this before, is that the sexual cycle also begins with aggregation, but under these weird conditions that I told you, which are the ones we know. We don't know what's going on in the soil. High intensity light, extremely high water tension, and so on. They form these zygote-like structures called the macrocyst. Now, in the macrocyst, something strange happens because the macrocyst turns out to be one of the large number of, I beg your pardon, the zygote turns out to be one in the large number of cells comprising the macrocyst. Remember, they've aggregated, and maybe up to 100,000 cells have aggregated. But this zygotic cell now turns cannibalistic and eats up the remaining, whatever, 10 to the 7 minus 1 cells. So the probability of cell death is huge. So the only surviving cell from this process now is this zygote. Only cell which survives within the macrocyst is the zygote. And then, of course, it undergoes the second reduction division and the amoebae emerge again. So the probability of cell death is extremely high there, enormously high. In this case, which is the case that we are, in this case, which is the case that we are looking at, the probability of cell death is variable. A while ago, I told you that 80% was the proportion of spores, in which case the probability of cell death would be 20%, but it can vary depending on the species. It can go up to 50% in our experience. So the only point I want to get across is that cell death occurs as a byproduct of each one of these processes to varying extents. In some cases, we can quantify how much. In other cases, we cannot quantify, but say that it probably is likely to occur. Therefore, the probability of dying, and in some cases a very high probability of dying, is something that the individual cell must take into account as becoming part of this whole game. So there must be something in natural conditions which permits in the long run this sort of life cycle strategy to persist, to be maintained, in spite of presumably a very high to moderately high probability of cell death. Please keep that one. Yes, please.
A multicellular thing, right? Because that could be another example of a multicellular.
It is a multicellular thing to begin with, as I said there, because the cells have aggregated. So those million odd cells are all there, But under these strange conditions, one of them differentiates into a zygote. It actually happens by cell fusion, as you can guess. Because, so this two haploid cells fuse, so it's a strange kind of zygote, meaning that you don't have differentiated germ cells. That zygote is within this thing called a macrocyst, right? And it's there, everything is dormant. But when the time comes for it to emerge, the zygote turns cannibalistic. So it's roughly twice as big as the other amoeba there because it's diploid and eats up all the rest, then goes through a meiotic division and comes out. Right. So now please remember, we are thinking of what's happening in nature and trying to see whether we can fit in our ideas to this. The first thing to keep in mind, and most of what I'm going to say now comes from a whole series of very interesting genomic analysis studies which have come up with molecular phylogenies carried out by Maria Romellaro, Pauline Schaap, Gernot Glöckner, and a number of others working with them. And the broad summary of these phylogenies, which we believe are objective phylogenies, and I must say that this is a strong assumption that we make, meaning that they actually trace lines of descent. The broad conclusion from these phylogenies is that what we thought of as relatedness, what we thought of as likely ancestor-descendant relationships based on similarities and differences in morphology, and what we thought of as a transition from something which was simple to something more complex, the assumption being that something simple arose far back in evolution and something complex was an evolved trait, all of these concepts go out of the window. In fact, as I'll tell you presently, the same thing has happened with these studies on the phyllasterians, the same thing has happened with this study on the Volvocales. So molecular phylogenies have completely upended simple-minded pictures of how evolution works by gradual modification from something which is elementary to something which is more and more complex. Right, so that's the summary of what I'm now. On top of that, there are many other things. So one of them being many of these morphologies, so these are fruiting body morphologies, as I told you last time, are facultative, which means that they are condition dependent. In particular, the transition between this and this, as I said yesterday, so this is a large size fruiting body, this is a small size fruiting body, and you can immediately see that cell size is one element in this difference, but that's an obvious and fairly trivial difference. The more interesting difference is that in this large size fruiting body, you have division of labor. These stalk cells are all dead amoebae, the spore cells of course are alive. In this small size fruiting body, you have no division of labor. You have only spore cells and the stalk is an extracellular exudate. So this is rather interesting. In a small size, if you wish, all cells do something in the beginning and then all cells ending up doing the same thing later. So you have changes in the pattern of behavior of cells as a function of time. Here, in addition, you have changes in the pattern of behavior of cells as a function of space. And this is a theme which will recur during what I'm going to say now, which is that in elementary organisms or unicellular organisms, you can get patterns of temporal differentiation. Different things are done by the same cell at different times. In multicellular units multicellular groups composed of units of the same kinds of things, different cells can do different things at the same time. So it looks as if this temporal derangement, polychrony if you wish, is an important element here. But the main point about this slide is that the morphologies quite obviously are not intimately tied to the genotype because they can vary in this facultative fashion. Secondly, in nature it turns out that these cellular slime mold groups are often polyclonal. You can also find clonal groups, but you can also equally often, maybe more often, find polyclonal groups. And in a way that's understandable, especially if these ruminants and grazers are agents of, indirect agents of fed. Because as I said, they graze a large area of land, crop a large area of land, and everything gets mixed up in their stomachs and finally gets deposited as a mix. There's no way of segregating different units. These genetically heterogeneous strains that you find coexisting in nature, quite often you find them coexisting on the same speck of soil, by which I mean the smallest piece of mud that you can pick up from soil. And you can very often not just get different strains of a species from these soils, you can get different species and indeed different families as well. So there's a huge amount of microbial diversity right there, which you can see very easily with your eye, coexisting within a spatial area which is small enough for all of them to be talking to each other. That's fairly obvious because the spatial dimensions here are probably of the order of a millimeter or half a millimeter, not maybe 100 microns, 1/10 of a millimeter. So you can ask how they talk to each other by bringing them to the lab and putting them together, forcing them to co-aggregate, which they do with more or less ease. So some of them do it very easily and form chimeric slugs and chimeric fruiting bodies. Some of them form chimeric slugs but then bifurcate into two fruiting bodies. Some of them bifurcate very early, and some of them actually give you bifurcating slugs. I don't know whether you can see that here. So they come together, but the slug itself shows segregation. So there are different levels of self-non-self compatibility. And the point of interest for us is that there are many strains which show this kind of behavior, which seem to be perfectly compatible, even though they are genetically different and therefore presumably have different reproductive interests. Right. So you can study these things when you bring them together and carry out the same kind of experiment that I mentioned earlier, which is to bring a number of cells together by counting, how many cells you brought together, how many of each kind, and having brought these two different genotypes together, you can ask, what are the relative proportions in which they succeed to become reproductive, succeed to become spores. So this is a measure of relative fitness in this kind of thing. And you can get all kinds of distributions in this thing. So within the same strain, so this is within a clonal population, you can get this kind of a distribution. So it's pretty much modal, if you wish, with some amount of error there. Between species, you get a U-shaped distribution, which means that generally every cell in a fruiting body is either of species 1 or of species 2. Very rarely you have something intermediate. On the other hand, between strains of the same species, you get something rather interesting, which is probably the same sort of average outcome, but a very noisy outcome. So these are things which are compatible with each other superficially. They come together, they build chimeric slugs and chimeric aggregates and chimeric slugs and chimeric fruiting bodies. But what they do after that depends on intense competition. So you see there's something rather interesting going on here. On the one hand, they seem to talk to each other and come together and build these communal structures. On the other hand, having come together, they seem to compete with each other intensely for reproduction. So you can see immediately that whatever explanation you have for their coming together, after coming together, what happens must have an element of reproductive fitness and relative reproductive fitness put into it. I'll wind up soon. Now, when you do this, and again quantitate outcomes, you get the following result. I just illustrate one slide. The result is that the level of incompatibility in terms of their coming together or not coming together seems to have a lot to do with differences at different genetic loci, and in fact a number of those loci have been identified. But having come together, right, whether there is what's called reproductive skew, which is a high asymmetry in who contributes to spores and who doesn't contribute to spores, that doesn't seem to depend on genetic relatedness. So you can take two strains which in terms of DNA fingerprinting seem very similar to each other, but show extreme reproductive skews. You can take two other strains which seem to be very distantly related to each other, don't share too many genes in common, but have hardly any reproductive skew. Therefore, they in some sense are much more friendly towards each other, you might say. So basically speaking, there seems to be a D-link between genetic relatedness here and levels of cooperation. If there's a link, it's not an immediately obvious link. Right. Secondly, these same phylogenies that I told you about have shown, and I've mentioned this already, that phylogenetic position in terms of complexity of form seems completely unrelated to phylogenetic position in terms of changes in ribosomal DNA sequence, which we think is objective. So organizational complexity seems to have give us no clue to phylogeny. Secondly, equally interestingly, if you look at the genomes of these amoebae, you find that a great many of the genes that they contain, which are believed to be essential for multicellularity, essential for coming together, are already found in their unicellular relatives. And when I say genes essential for multicellularity, what I mean is this: these are genes which, when mutated, don't do anything to the cell during its unicellular phase. So the cell lives as a normal cell, feeds as a normal cell, grows as a normal cell, divides as a normal cell, but is unable to become multicellular. So the assumption is that these are the genes whose evolution may have been responsible for the transition from single cells to multicells. But 80% of those genes, as you can see, are already there in other unicellular relatives which never become multicellular. Therefore, adding to the weight of feeling that it was not the acquisition of these genes that fostered multicellularity, but that multicellularity came about for whatever reason, taking advantage of the pre-existence of those. In fact, many of them are also found in non-amoebozoan species. Genes, regulatory mechanisms, and morphogenetic pathways as well are shared with the so-called higher metazoa. And I have written down a list of those genes which have been published: integrins, Wnt, STAT, tyrosine kinase phosphorylation, homeobox genes, regulation by non-coding RNA. So the kinds of things that are usually believed to be essential for coordinating multicellular development in the higher plants or higher animals are found here in these organisms already. And many of these genes are present in their unicellular relatives who never become multicellular. Same kind of thing, which shows an overlap between genes in Dictyostelium and genes in other representatives of other supergroups, which are constitutively unicellular. Now, I'm coming to the close of what I want to say, which is to sum up the picture that is building now and indicate what Volvox and the phalasterians have to say, which reinforces. First of all, you can get aggregation without building a fruiting body. You get this in this related amoeba called Hartmanella. You can get a fruiting body without aggregation. This is extremely interesting. This is an amoebozoid called Protostelium, which is a single cell, a single amoeba, and it forms this supposedly multicellular structure. Obviously, it's not a multicellular structure, but it builds a fruiting body, which means it secretes a stalk and uses negative pressure to climb up to the top of the stalk where it forms a spore. So what I called this engineering problem earlier of building the fruiting body is already present in a unicellular organism. Now there's no evidence that this protozoan, this protostelium, was ancestral to the Dictyosteliates, but this is rather interesting. You have other protozoans which are believed on the basis of molecular phylogeny to be ancestral, which sometimes show this sort of single cell fruiting body. So it appears as if the engineering problem had been solved in an ancestor. That solution clearly was not carried over into this descendant, but the capability for carrying out the solution was in some way, and that seems to be embodied in this. You can get differentiation. This I've already mentioned, and division of labor without cell death. So you can get a stalk which is acellular. Stalk which is acellular. Now, so very quick remarks on the other two things. So the choanoflagellates, which are at the top of this picture, are the closest unicellular relatives of us, of the metazoa. So this is the closest unicellular relatives of the animal clade. The phalasterians are, if you wish, next closer, and then the ichthyosporians were next closer. The picture from these three groups, especially from the phalasterians and Capsospora, which I mentioned a while earlier, is that these so-called multicellularity genes that we spoke about a while earlier are present in them and also in their unicellular relatives. So once again, in that lineage too, you didn't need major acquisition of new genes to get multicellularity. Secondly, this is very curious, which is also interesting. Many of the genes which we had believed hitherto were new to multicellular organisms turn out to have been genes which were lost in an ancestor and then regained later, which means the genes were there. The genes and the gene products probably were doing something else in the immediate ancestor, right? And then their original function was regained in their present-day descendants. So basically, the unicellular ancestor of animals had a gene repertoire associated with differentiation and multicellular animal development. So you had the genes, you had the networks, but you didn't have multicellular development because these are obligatorily unicellular creatures that we're talking about. But again, the thing that I said earlier, which is that you have spatial patterns of differentiation seen now in multicellular organisms, which seem to be prefigured in these unicellular organisms in terms of temporal patterns of differentiation. So these are just a listing of the sorts of genes which have been found in Capraspora and Gynoflagellates. Volvox now. This is really a textbook example in the sense that it is used in textbooks as an illustration of how multicellularity could have evolved, and it shows a very nice series of graded complexity. And the graded complexity that is shown in books is Chlamydomonas, which is a single cell, some of these intermediates, which are Gonium and Eudorina and Pandorina, all the way up to Volvox, which is probably up to 1 million cells, certainly 100,000 plus cells. So these are different members of the Volvocales, which show different levels of complexity, also different sizes, and the relatively uncontested assumption until recently was that in the course of evolution you had something like the Gynoflagellates, then something like Gonium, something like Eudorina or Pludorina, and finally something like Volvox. And what is also interesting is that Gonium, which is multicellular, Chlamydomonas, of course, is just one cell. So there is no question of a germ-line soma distinction there. Everything is a somatic cell because there's only one cell, then everything becomes a germ-line cell. Gonium shows the same feature. All these cells keep swimming, performing somatic functions because they are swimming in the search for food. So all of them keep feeding, growing, and once they have grown enough, presumably all of them become germ cells, and you can get reproduction as the next step. Eudorina, which is in between, shows something interesting. And here you have an almost mathematical rule. If Eudorina consists of 32 cells or fewer, you have the same pattern as in Gonium. Everyone is a somatic cell, and then everyone becomes a germ cell. 32 being 2 to the power 5, you can see immediately that that results from 5 mitotic divisions. But 64 or more cells, you start getting spatial division of labor. So you already have, within the 64 cell mass, some somatic cells and some germ cells. So you have the same kind of uncanny resemblance to size-dependent division of labor on the one hand, and secondly, temporal patterns giving way to spatial patterns once sizes increase. But the most stunning piece of information that we've got out from Volvox phylogenies is that this picture of going from simple to complex completely breaks down if you look at the real phylogeny. Well, yeah, this was supposed to be the kind of way in which the phylogeny worked. But it turns out now that when you do the molecular phylogenies, you find single clades which contain both a Volvox type of lifestyle plus a Gonium type of lifestyle, both a Gonium type of lifestyle plus a Eudorina type of lifestyle. So the phylogeny in terms of traditional species names, traditional species identifications based on what they look like, right, and based on how their developmental course went, that is completely muddled up, totally muddled up. Once again making the point that if your molecular phylogeny is actually showing nearest neighbor relationships in evolution, and therefore genetic, in some sense, genetic nearness in evolution, that has nothing to do with the way lifestyles change, morphologies change. So grades of organizational complexity are completely different from clades of relationships. And you have other bizarre things. In fact, this is a point made by Volvox workers, and Kirk does it in this paper, which is a single mutation in Volvox carteri changes it, a single point mutation, changes it completely to a form and a lifestyle, which if you had seen it for the first time, you would have said, this is not Volvox, this is Eudorina. Remember, the single cells can't be distinguished very easily. All of them look like chlamydomonas, no, in a sense. Okay, in a single mutation involves power C changes it, so it could be called plurorina, and so on and so on. So, genus and species names identify grades of organizational complexity. These are traditional names, not clades of classes. Okay, so I end here. So the point here seems to be that genetic overlap and genetic similarity are not as strong grounds for fostering groupness, multicellularity in our case, and as I said, perhaps social behavior as one had thought of. And there are at least two interesting examples which show that, which is that even in large animals, you can get chimeras under the appropriate circumstance, which means aggregates of different kinds of cells. Here the aggregates are made artificially, of course. This is a sheep and goat chimera. And if you're lucky, these creatures can go to term. They don't look very healthy, but nonetheless, they've gone through embryonic development completely normally. And the idea is that these gross genetic differences between these two cells haven't prevented the cells from talking to each other and organizing this multicellular form, because whatever signals, adhesion factors, and so on that they needed were compatible. This is a chick-quail chimera. Here the two, again, artificial aggregation followed by hatching in the lab. The divergence was even longer ago. So this brings up this question which Stuart Newman has tackled in many of his papers, which is that maybe, and this becomes completely speculative at this point, but it's interesting speculation, maybe there is a solution to the old problem of which came first, the chicken or the egg, and the solution is that the chicken came first, meaning the multicellular organization came first. And then secondarily, via competition for entry into the germline, right? Some of these, so this is the picture that we are looking at and have been looking at here, this one. All of these, of course, are the things that we know now: fertilized egg, embryo, whatever, blastocyst to maybe blastula, gastula, and then embryo, and then adult. So the question is, could this kind of developmental strategy have given way to any one of these kinds of development strategies? And he argues that it could have, because this, which may have become stable in certain situations, as for example in the unicellular organisms we've looked at, may have given way to this by way of reproductive competition between the cells within the aggregate, and the one which ended up as the winner in this competition became what you now call the egg cell. Of course, now you're suddenly confronted with this problem of sexual reproduction, which you have ignored all along, and figuring out how you have this transition from a haploid state to a diploid state. But I want to leave you there. That's the end of my talk. Basically, what I've tried to get across is that there are organisms around now which go through a unicellular to multicellular transition as part of their normal life cycle. B, many of these organisms seem sufficiently ancient that you can think they might offer you interesting clues for the origin of multicellularity. Remember, they occur in essentially all of these supergroups that we looked at. I said that they didn't occur in the archaeplastids, but even there, it's rather interesting. If you challenge chlamydomonas with a predator, which is usually a ciliate, it turns out that they gather together and form aggregates. Saté and Durand showed this recently. So to all outward appearance, they're like one of these other creatures, except of course they're chlamydomonas cells which have got together. They don't change in any way. But they do come together, they do aggregate. And what's interesting is that in this aggregation, they make no distinction between clone mates and otherwise. So they work with genetically heterogeneous samples, so that plays no role. So that's the second point, to show that this might offer interesting clues. And thirdly, to say that probably by looking at the broad features of coming together and pattern formation, we might be able to get some clues to the origin of multicellularity, rather than by trying to explain multicellularity by the acquisition of new genetic functions, which resulted in small advantages at least. Thank you.
When this slug comes together, how do they decide on the axis, like where will be the start and end? And if they move, like, is it purely chemotaxis? And how, like, this computation, like, how do they decide where to go?
Okay, there were three things there. The first question was about the axis. What was that?
Yeah, when they come into this mass slug, how do they decide where will be the...
Right, okay. So when they come into this mass, it turns out that there's a great deal of sorting out, very similar to the sorting out that we saw earlier. And this depends on differential adhesiveness, as also on differential chemotaxis, right? Which is there, inherent in the system, even if you have a clone of cells, right? So you remember that I talked about heterogeneities prior to aggregation? These heterogeneities are correlated with differences in many of these other factors, right? Which lead immediately to sorting out. So differential adhesiveness can give you one mechanism for sorting out. Is that okay or no? If different cells have different levels of sticking to each other, then they can separate. You can get a kind of phase separation. Okay. And the second thing was, you said, does it move by chemotaxis? The slug does not move by chemotaxis. Chemotaxis is the process that brings cells together. As far as we know, the slug does not exhibit any chemotaxis. On the other hand, it exhibits strong phototaxis and a kind of negative geotaxis, both of which make it come to the soil surface.
But where does this computation come from? Like, is there any like leading cells that...
Sorry?
Like, how do they come to this common decision together?
Okay. So the understanding is that after starvation, this whole series of reactions are initiated within single cells, and these reactions consist of A, acquiring gradually these adhesiveness properties, B, of making and releasing cyclic AMP, C, of developing this positive feedback mechanism which doesn't exist earlier, and these are all properties that arise in single cells individually. So they are pretty much the same in all cells except for the heterogeneities that I mentioned, and that's enough to make them come together. The oscillation comes about because of an interesting combination of positive and negative feedback. The positive feedback is that cyclic AMP outside the cell can bind to the receptor and trigger more cyclic AMP synthesis. There are two negative feedbacks. They're rather interesting. The simpler one to understand is there are phosphodiesterases which are induced by cyclic AMP, right? So when the cyclic AMP level gets very high, the enzyme activity gets very high and the cyclic AMP level crashes down. But the more interesting kind of negative feedback comes about because of receptor desensitization, right? So the receptors binding to cyclic AMP crosses a threshold, we think, we don't know whether it's a clear threshold. Then it turns out that the positive feedback loop is cut, right? And the cyclic AMP degrades again because of enzyme. Of course, it's also diffusing away, so. The cyclic AMP sent by one cell does two things. It signals to other cells to come to it. It signals to itself to set up this feedback loop, which initiate oscillations, and presumably also triggers other cells to get into the oscillatory path.
So I have a couple of questions. First is for Dictyostelium. When the cell starts starving, each of them is secreting cyclic AMP. So where do they decide to finally aggregate to? Wherever the highest concentration of cyclic AMP is?
Yes, which in turn means wherever you have a locally high fluctuation of the concentration of cells.
Okay. And in case of Chlamydomonas, when they're challenged with, say, a predator, and they just aggregate and don't do anything differently, I'm wondering what the advantage of aggregation in this case is.
Sorry, I should mention that.
Move forward.
The ciliate can't swallow them anymore.
Oh.
They're too big.
Oh, I see.
Exactly. So they survive.
I see. And also the point mutation that could change Volvox into something like the unicellular.
Not change really, you know what I mean. If you hadn't known that that had happened and you were chancing upon this for the first time, right, you would call it Eudorina, something or the other.
But is it known what mutation it is?
Yes. Unfortunately, not known to me at this moment, but I can look it up and yes.
Thank you.
By the way, there's an amusing thing related to that, which is there was a massive collection of natural isolates of many of these species made in Australia, if I remember correctly. And this was in particular a hunt for Pludorina, so you went by traditional taxonomic criteria and identified a number of them. Later, when molecular sequencing was done, it turned out that these were all misnomers. One of the Pludorinas was really a Volvox, the other one was really some, Gonium, other was something. So there's this whole big debate now, certainly in microbial taxonomy, about what to do with these old names that we have. And that's as true with Dictyostelium as with the others.
Hello, can you hear me? This side. Yeah. So my question is maybe something different from what you talked, but in similar lines is the symbiotic association, right? As in, we know that eukaryotic cell evolved from a prokaryote after engulfing other cells, right? So you talked about temporal differentiation wherein cells together perform one type of work than another type of work. But a model wherein they two different organisms cooperate, right? Such kind of model which I didn't, I was, so I wanted to know some work on those lines wherein organism developed after the symbiotic association, and which will be completely different model compared to the other model that you suggested.
Oh, hang on. So within the theme of multicellularity, what you're saying is that multicellularity could be maybe an obligatory byproduct of symbiosis. Yeah. Is that correct? Is that what you're saying? Well, in a way that happens in some cases, for example, in some of these phylasterians, it turns out that this aggregation, which I've been talking about, please remember, the situations which I described in detail had to do with these amoeboid organisms which came together, built fruiting bodies with or without division of labor. And you find these in at least 6 of the 7 supergroups. So that's the situation that I looked at. I also mentioned that you get multicellular aggregations, multicellular structures formed in the phylasterians, in ichthyosporians, in Capsaspora, and so on, right? In the choanoflagellates, and so on. Now, there it turns out that the route to multicellularity need not be starvation. So quite often, these choanoflagellates, which feed on bacteria, gather together where there are a lot of bacteria. So in some sense, it's a food-induced coming together, and then they form this multicellular mass. So now I don't know whether you want to call it.
That food is not complementary, right? As
exactly.
So
I was going to say.
Find complementary things. So what makes them come together?
People who work on bird flocks, which is a form of multicellularity, and many cases this can also involve multi-species flocks. So these birds flock onto fruit-yielding, fruit-bearing trees, and there you can see the symbiosis.
They are quite similar organism which is coming.
Pardon?
In bird flock, similar kind of
birds of different species, even multi-species, they can flock on a given tree. I'm not saying that they are collaborating, but this flocking helps the bird, presumably finds a roosting place, and by having large numbers of birds in one place, you get a better risk of predator avoidance. And the fruits have the usual obvious benefit. They eat the fruit and deposit the seed somewhere and so on.
I guess there should be some, like if organism is very big, right? They won't symbiote easily. If organism is at the like multicellular level where they are like 10 cells, 20 cells, or
no, you get symbiotic associations between large organisms as well.
Okay. Just like humans do have, yeah, true.
Hello, yeah. So when we see the slug part, and there is this spatial differentiation between pre-stalk cells and pre-stalk. So so it seems like the differentiation was before the slug motion, and that the differentiation could be like for differential cell migration or stuff like that. So I would like to know if there is some work about how does this initial differentiation occurs, if there's a spatial pattern, like, for example, pre-stalk cells being in the center of the amoeba aggregation, or something related more to space in the beginning. So.
That's really an interesting point. The general belief is that there is no such relationship, but there are some experimental findings which badly need to be repeated.
Sorry, what?
Some experimental findings which need to be reconfirmed, but they're rather interesting. So for example, there's one set of findings which says that the cells which begin to signal, initiate the process, ultimately become spores. That would be rather nice if that was true.
Oh, yeah. I guess I'm rather surprised that you haven't said anything about quorum sensing, because, well, before this talk, I would have thought that the difference between multicellular and unicellular things is the aggregation, but given the talk from yesterday and yours, it seems that aggregation is just a thing that cells do in the same way that unicellular organisms quorum sense. So is it that we're just calling things unicellular that aren't so uni? Like they're coordinating among themselves, but they might not be like physically attached. Is that?
Yeah, I mean, I'll go along with that way of thinking, yes.
So you showed some spiral waves, the C-amp oscillations. So that is the pseudocolor plot was the C-amp concentration.
Sorry?
C-amp concentration, the pseudocolor plot you showed was a C-amp concentration. And the center of the spiral wave, is that a single cell which is giving out the signal, or is it?
Okay, that's a very interesting question. Perhaps you could discuss it later in detail. But the center of a spiral wave can also be a hole.
Yeah, it's singularity.
Exactly.
Vidya, so towards the end of your talk, you're alluding to chimerism, and you're giving two examples of artificial chimera that were generated in animals. So my question was, or more comment, I wanted you to extend a bit or comment a bit on the fact that you're showing these two examples that are artificial, but we know that this situation is not that rare and is in natural organisms, in animals in particular. You want to comment on this?
Well, briefly, maybe you can comment more. I'll tell you the two cases which come to mind right away. Ascidians do this a lot of the time. They show fusion, so you can even get adults fusing with each other and developing chimeras. And there's very nice work being done, having been done and is being done, I think, by Rinkiewicz, and you first pointed it out to me, which looks at competition for entry into the germ line after chimera formation in Ascidians. And yesterday, of course, Stuart showed us this remarkable finding in planarians, which seem to be quite often obligatory chimeras. Any other cases you know?
Yeah, so that's two examples, including the example that Stuart presented yesterday that I didn't know about. It's actually a situation that is also observed in mammalian species, including in humans. And I just showed to Zorana an example of a paper from 1979 in Nature, where so it was the very beginning of genotyping at the time, and there was a case of a mother who was genotyped, and also 4 children. Obviously, it should all match.
Right, you're right.
And in fact, there was no matching between any of the 4 children and the mother, which is very, very, very surprising. Of course, the random exchange of the 4 children on 4 separate occasions at the maternity was also excluded. And the explanation is the mother is a chimera, and there is no symptom. There is no disease associated to this situation. It was found just completely by chance. And the explanation is this person is a chimera, and the cells that were used for genotyping are coming from her blood. And of course, the cells that gave rise to her children are not from her blood. And this is the explanation. So chimeras are probably much more frequent than we think, even in marine species. That's all.
Thanks for pointing that out. I believe that there are many other cases which human geneticists have come across where you get things like double fertilization, followed by chimeric development. In other words, simultaneous ovulation of two eggs, both of which get fertilized, and then the zygotes join up together, and apparently there are examples of that as well.
You have told us about how multicellularity can be defined in one fruiting body, but have you seen a more the interaction between fruiting bodies, and maybe giving us some kind of ideas about, I don't know, multicellularity can be explained studying one fruiting body, but it can be explained also if you see the groups of, or one group of many fruiting bodies. I don't know.
That's a very interesting question. I was just thinking of something while you were saying this. To begin with, I don't know how to answer that question, because I don't know if there's one well-known interaction between fruiting bodies which results to spacing. It turns out that while fruiting, these, at least the slime molds that have been worked on most frequently, they release ammonia, and ammonia is a repellent gas. So the fruiting bodies make sure that they stay away from each other while fruiting. So that's one kind of interaction. But what that has to do with multicellularity per se, I'm not sure. And whether there are other interactions, I don't know. There's something else which might interest you. I don't know whether this is in line with your question. This is a long unsolved problem in some sense. There are speculations, but I think we still haven't got our hands on it. The problem is the following. If you plate these cells on a petri dish in the lab, you get, and finally you get fruiting bodies, you get a certain density of fruiting bodies. Right? Now that density turns out to be pretty much invariant over a huge range of cell densities. In other words, the more cells there are, the larger any given fruiting body is. But that doesn't mean a larger number of fruiting bodies in a given area. You get the same number as before with the same spacing as before. So the territory size, if you wish, of an aggregate seems rather insensitive to the number of cells who are in the aggregate. That's rather, it's almost as if you had cities or villages decided initially, and whatever the number of people there were divided themselves among those villages with the fixed spacing. It's not known how that's done, but presumably that also involves some form of interaction. I don't know.
So, can you hear me? Yeah. So when they become, when we starve them, they come together as aggregates, right? So when is the decision final? If we give the food back to them, when will they, will they again become single cells? Till what stage they can revert?
Very interesting. Someone said once long ago, these cells never die until they're really dead, which means all the way up to the very end, until terminal differentiation occurs, you can give them food and they go back.
So is there any way that we can track if same cells become the stalk or spores every time?
Very interesting question. Okay, here's a finding. Yama, you remember who did this together with Takeuchi? I forget the... Someone and Takeuchi did this experiment. So you had these cells come together, form stalk and spore, right? Now there are tricks by which you can get amoebae to come out of the spores even though there's no food. And one of those tricks is heat shocking. So you can get amoebae, of course there's a fixed number, they don't grow or divide anymore. You can get amoebae which have gone through a life cycle and have become amoebae of the next generation in the absence of food. So if you take amoebae which have come in this way from spores, and you take other amoebae which have not come from stalks because the stalks are dead, but which you generate from the stalk cells in this kind of way by disaggregating them. And then you mix them. Turns out that the ones which came from spores form spores again. Follow? And the ones which came from stalks form stalks. So there's some form of epigenetic, let's use the word inheritance here, but its basis is not known. This pattern breaks down if you allow them to feed in between and go through mitosis. Then everyone forgets what their background was.
So let's thank Dr. Nanjundiah for the insightful talk. And